This video I will discuss about the purification of the colloidal solutions. In the previous video, we discussed about the preparations of colloids. Now let me discuss purification of the colloidal solution. How do you purify? After the preparation, they will have a certain amount of impurities in that. We have to remove that impurities present in the colloidal solution when they are prepared. The first method, let me see in this case, then why the purification of the colloidal solution is required generally if you observe. Colloidal solutions when you are preparing, generally they are containing X amount of electrolytes and some other soluble impurities. Why? Because in the peptidization method, by adding some enough amount of electrolyte, you are producing the, isn't it, colloidal solution from the precipitation. So you are adding what? If you add excess amount of electrolyte, it may have some excess amount of electrolyte. You have to remove that one. And whatever that precipitation may have sometimes soluble impurities also. So we have to remove that one. While the presence of trace amount of electrolytes is essential for the stability of the colloidal solution. So having a little bit amount of electrolyte is okay. But have more amount, that will what may convert that into coagulation. Coagulation is nothing but reverse to peptide. Reverse to peptidization means conversion of what? Colloid into what? Precipitation. Okay, so that may happen, sir. If you have excess amount of electrolyte, if you add, what happen? That whatever the colloidal solution again will convert into precipitation. So that is called coagulation. We'll discuss later. Now, therefore, it is definitely what happened. We have to reduce that concentration of the soluble impurities to required minimum amount here. So, the process used for the reducing the amount of impurities to require the minimum amount, you are calling as a purification of the colloidal solution. Purification of the colloidal solution is carried by different methods. The first method we are taking as what? Dialysis method. The first method is what? Dialysis method. Now, what is this dialysis process? Let me see. Now here, in this process, we remove the dissolved substances, okay, from a colloidal solution by using which method beta? Diffusion through suitable membrane. Diffusion through suitable membrane. Now, what is this meaning here? Diffusion through a suitable membrane means what actually? Let me discuss about this one. Diffusion is the net momentum of substances. The substance may be atom, may be ion, may be molecule, from a region of high concentration to low concentration. Okay, the movement is move is taking place from high concentration to low concentration. This is called what? Diffusion. Let me discuss in detail again. Now I have taken the membrane, which may be what happened, animal membrane or parchment paper or cellophane sheet also we can take care. Through this only the diffusion will take place from high concentration to low concentration. The diffusion moment can take place by the animal membrane or bladder, we can call this one. Animal membrane or parchment paper or cellophane paper is a medium through which what happened, the diffusion takes place. Now, it is a selective barrier which allows the momentum of ions or small molecules and retain the colloidal particles. Now, what is the function of this cell membrane here? This cell membrane, isn't it, having small minute holes over on it. Now, therefore, it allows the momentum of ions which are having very small in size, very small in size. And the large size, like colloidal particles are large in size when you compare with what? These small particles. So they retain over here only. The small particles will flow through what? Cell membrane. So this is the process actually happening. So the apparatus we are using in this dialysis called dialyzer. Isn't it? Dialyzer we are using in this process. So working principle of dialysis, how it takes place. If we take this experiment here. Now here, this is the membrane, the dialysing membrane we are using here. It may be what happened, animal membrane, otherwise what happened, parchment paper it is. Okay. Now, this inside you are having what happened, you are having something colloidal solution. So you are having here colloidal solution, which you are taking in the vessel actually. Okay. This is the vessel you are having in this vessel, what? Colloidal solution you are having. Now, if you observe what happened, this particle, this big one, green particle, The large particle is a sole particle, nothing but a colloidal particle. Whereas a smaller one is a crystallite. Crystallite means a salt, maybe a salt particle, which is having less size. Now, when you start the dialysis process by something what happened, having in the water, when you put this container vessel in the water, the dialysis will start now. Now, when you observe what happened, what is diffusion? Movement from concentration to low concentration, high concentration to low concentration. So as water continuously flowing, the molecules and ions will diffuse to the membrane into the outer water, and pure colloidal solution will be left behind in the bag. Now, what is the meaning if you observe here? This is the membrane. This membrane, observe here, this membrane will allows, this membrane will allows only small size only, because the small minute holes are there. So to the small to minute holes, the large size of colloidal particles cannot pass. Which can pass better? Only what happened? The minute size we are having, because the membrane is made upon by the minute size, and the minute particles only can pass through what? This membrane here. So definitely what happened? The small particle, this one, they are what? Passing through. And why they are passing into the water? Because diffusion takes place from high concentration to what? Low concentration. What is happening? From high concentration to low concentration, whatever the impurities are moving. That salt, isn't it, is moving. The electrolyte excess amount is moving into it, isn't it? Now they can be removed from the water later. So this process you are calling as what? Dialysis process here. Correct? Next. Purification of colloid solution by using second method, electrodialysis. Now, what is the difference between the electrodialysis and dialysis? So dialysis is a slow process, better. It takes place very slowly. So to speed up this method, we have taken what? Electrodialysis method. The method we are obtaining here, electrodialysis, by keeping this dialysis in the electricity. Right? So let me see this one. So by applying electrical field, okay, we are making this one into very fast. The process named then electrodialysis process. Now working principle, let me observe. Everything is same experiment it is. Everything that we are taking, the operators are same only. No difference in that. To here it is same. Now here if you observe, I'm applying what here? Positive charge. Here it is negative charge we are applying. Right? Positive and negative charges we have applied. Two electrodes, positive electrode, negative electrode, applying to what? Dialysis. Now, because of this dialysis here, what is happening? Present in the colloidal solution migrate out to the oppositely charged particles. Now, here, these are again the positive and negative again are what actually, isn't it? Salt particles. For example, Na plus and Cl minus, which you are calling as impurities. You are removing the salt particles like NaCl impurities. The green part is nothing but colloidal particle. So now, when you have your positive charge is there, the negative charge readily move to what? Positive, and the positive charge readily move to negative, isn't it? So you are speeding that whatever this dialysis method by using what? Your electrolytic method. So they come down here and they can be easily removed. So because of these electrodes, positive, negative electrodes, ions are coming outside to the semi-membrane, that's whatever the cell membrane coming outside, so that making the process what? Very fast, right? So this is called what? Dialysis method. Now let me take some process of removing dissolved substances from a colloidal solution by means of diffusion, called always what happened? Dialysis method. The process of dialysis can be made faster by applying purification. Let me take second type of purification now, that is ultrafiltration. After the dialysis, electrodialysis, the third one we are taking ultrafiltration. Now what is this ultrafiltration here? It is a process of separating the colloidal particles from the solvent and soluble solute present in the colloidal solution by specially prepared papers actually, specially filtered papers, which is permeable to all substances except the colloidal particles. Now, in the previous process, what happened? So in the previous process, that not 100% we cannot prepare purification. 100% we can't purify that. In that case, what happened? We are separating whatever the colloidal particles from whatever the solvent, and the soluble solutes which are present in the colloid solution. These soluble salts can be, isn't it, removable, isn't it, by using what? Specially prepared filter papers. So these are what nothing but ultrafiltration. Let me see this one. Now let me take here what? An ordinary filter paper. Ordinary filter paper present in the chemistry lab. Generally, we are doing for the drying that one. A filter paper you are taking, ordinary filter. Now this is a colloidal solution and colloidal particles. This is a green part is a colloidal particle, sole particle, and the blue part is the impurities we are taking. Crystalloids we can call this one. This is made upon by the salts. Now colloidal particles can pass through ordinary if you take ordinary filter paper here, both can pass because ordinary filter paper have bigger size, bigger holes it will have. So through the bigger holes, what happen? Both particles, sole particles and crystalloid particles, both will pass. So there is no use for us. We can't separate by using this. By using what? An ordinary filter paper, it is impossible to purify what? Colloidal solution. Now what we are doing here, in the place of ordinary filter paper, what we have to use. So now take again that ordinary filter paper and dip that ordinary filter paper in a mixture called nitrocellulose. It's a very important term. Nitrocellulose. What is nitrocellulose in examination? They ask actually. What is the meaning of nitrocellulose? They ask. The meaning of nitrocellulose is 4% of the colloid ion. What is the meaning of colloid ion? They are asking. Colloid ion they are asking. Colloidin ion is nothing but it's a solution mixture of what? Nitrocellulose. Nitrocellulose plus what happen? Alcohol. In the place of alcohol, you can take ether also. This mixture is called what? Colloid ion. In this, we have taken 4% of nitrocellulose. That means 4% is nitrocellulose and remaining will be what? Alcohol. The solvent actually. Alcohol is a solvent. This one is a solute. So what is colloid ion? 4% solution of nitrocellulose present in the alcohol or ether. Now dip this ordinary filter paper in this solution. So what happens when you dip? What happens? That big holes, whatever the big holes are accumulated by this solution. So by the ultra filter paper you are going to get now. Soaking the filter paper in the colloidal solution. Hardening the soaked filter paper with formaldehyde solution. Finally, what happened? Drying. So in this method, by using these three methods, we get the ultra filter paper. First of all, ordinary filter paper, you have to soak in where? Colloidal ion solution for certain time. Then what happened? You have to make that one into hard by that whatever the soaked filter paper, you have to harden that one by using what? Formaldehyde solution. 40% of formaldehyde solution. In that you have to dip that. When you dip, what happened? The paper will get hard. And after that, what happened? You have to dry that one. Then the paper you got, that ordinary paper, now converted into what here? Ultra filter paper. So now when you use ultra filter paper, isn't it? Now we use what? Ultra filter paper. What happens here? Now I have taken ultra filter paper. In the place of ordinary filter paper, I have taken ultra. Now what happens here? So when you do what happened? By using high pressure, when you apply some high pressure, what happened? Because of the gravitation, which particles are coming down here? That crystalloid particles, impurities are coming down, and whatever the sole particles, colloidal particles are remaining in the medium only. So this is called what? Ultra filtration method. The important thing is that nitrolim solution is very, very important. Ultra filtration is carried by which one? Specially prepared filter. We have to use specially paper. Colloid ion paper. We can call colloid ion. Colloidon means what? 4% of cellulonitrose plus remaining will be what? Alcohol or ether can be called as colloid ion. That is very, very important for examination point of view. So with this, what I have completed what? That purification of the, isn't it? Colloids also completed.